Yes, ma'am, you are audible.
Good evening, madam.
I'm Dr. Vijaya Padma. Good evening.
Vijaya Padma, madam. Good evening.
Bharat-
This is Professor
Yes, sir.
Ramara from Geetam.
Good evening, sir. Am I not audible? Am I not audible? Yeah, yes, sir. Am I not audible?
You are audible, ma'am. You can proceed with your presentation.
Yeah, yeah. Okay, sir.
Okay.
Yeah, yeah, yeah. Okay, sir. Sir, let me present my work. We'll discuss later, sir. Okay. So today I'm going to present my work on reversal of cisplatin resistance by treatment with neferine or isoliensinine, either alone or in combinatorial regimen, along with cisplatin in cisplatin-resistant colorectal cancer cells. So, as all of you are aware, the colorectal cancer is a highly invasive
Manas,
cancer, prevalent among aged individuals. Hello? Shall I continue?
Yes, ma'am, please continue.
Yeah. So generally available therapy options are conventional therapy options include surgical interventions, radiation, and also chemotherapy. But still the mortality of patients is still very high because of the resistance, which is generally used chemotherapeutic agents are 5-fluorouracil and cisplatin, carboplatin, and things like that. But still, CRC patients, if they, it's not because effects along with the poor clinical outcome, their usage is limited. So cisplatin chemotherapy to the colorectal cancer cells is severely associated complications like and things like that. So, and also acquiring resistance therapy is also a major problem. Therefore, there is a need for a serious alternative this thing. So, cisplatin resistant stem cells are generally arise due to continuous exposure to cisplatin treatment because of the overexpression of the drug resistant proteins, that drug transport proteins which are associated with the, we generally call them as multi drug resistant proteins, which are associated with the membrane, cancer cell membranes, which fuse afflicts the drug, which won't allow the cisplatin to get accumulated in the cell. So this overexpression of this is one thing, and majorly with cisplatin, it is actually drug repair, because usually cisplatin causes cytotoxicity by causing cross-linking, DNA cross-linking. So cytotoxicity is majorly because of damage to chromosomes or cross-linking of the chromosomes. So because of the DNA repair, extensive DNA repair, to drug cisplatin drug happens in the, and also people have noticed that a lot of stem cells are because of the generation of stem cells also. So the stem cells characteristics also, this continuous exposure to cisplatin stem cells or accumulation of the stem cells, which also proliferate and their number increases. So the stem cell characteristics are because of the presence of these markers, stem cell markers. They can be identified by the expression of the stem cell markers, which include CD24 and CD44, which are surface markers. Apart from that, also enhanced repair proteins, DNA repair proteins, such as ERCC1 and also cell survival proteins. So enhanced expression of all these things together results in accumulation of stem cells, cancer stem cells, which results in drug resistance and also poor therapeutic outcome due to cisplatin treatment. So there is a need for alternative therapy. So here we tried using neferine
Is
and isoliensinine.
presentation mode.
Hello, are you not able to see, sir? Sorry.
It is not in the presentation mode.
It is. If screen is visible.
No, ma'am, it's not visible now.
Okay. Is this visible now? Is the slide visible?
Yes, ma'am. Yes, yes. You can continue your presentation.
So yeah, see, the cisplatin resistance is due to continuous exposure to cisplatin, which lead to accumulation of stem cell characteristics in the cisplatin induces stem cell characteristics. DNA repair and survival protein expressions. Treated cancer cells. So we have tried alternate therapy by using neferine or isoliensinine, either alone or as adjunct. That means along with cisplatin, we had given either neferine or isoliensinine treatment for chemosensitization of cisplatin-resistant cancer cells. So we have found that neferine and isoliensin, both of them treating alone with either of these things had shown they both have anti-cancer effects. That means they have chemosensitizing effects against drug-resistant cells, cisplatin-resistant cancer cells, as well as we could even when they are given in combination with cisplatin, that means neferine with cisplatin and neferine with isoliensinine, either in the combinatorial regimen or alone. Given alone also, they had shown chemosensitizing effects against the cisplatin resistant cancer cells. So this we have chosen these alkaloids. These are basically neferine and isoliensinine are bisbenzyl isoquinoline alkaloids, which are derived from the lotus. Lotus is commonly Dilumbo nucifera is its scientific name, commonly known as lotus. So these the alkaloids are derived from the lotus embryo. We have isolated them from lotus embryo. Have already been reported. Even in our lab, we have reported with doxorubicin. This is a study where we are, today we are going to discuss the cisplatin resistance. In combinatorial along with cisplatin. So this is our aim of the study is to evaluate the chemosensitizing efficacy of neferine or isoliensinine either alone or in combinatorial regimen with cisplatin in colorectal cancer cells, stem cells. That means resistant cisplatin resistant colorectal cancer cells. So the methodology includes first we have developed the cisplatin resistance in colorectal cancer cells. But then we have HCT15 cells we have procured from the National Institute of Cell Science, National Center for Cell Science, Pune. Then we have by continuous exposure to cisplatin, we have developed cisplatin resistant cancer cells. Then we have exposed them to treatment with neferine or isoliensinine and determined the IC50 values. Then we have also then we have determined the ROS. We have got determined, and also intracellular calcium levels, mitochondrial membrane potential, and also the apoptosis induction was confirmed by SIM analysis as well as SIM and TEM analysis and PI uptake, propionimidate uptake, and cell cycle analysis. And also we have done early apoptotic apoptosis was also determined by NXC using flow cytometry. And then we have done the expression profile of the stem cell markers and the DNA repair proteins and all using mRNA, that is RT-PCR, and also we have done a Western blot analysis for all the apoptosis and cell survival and DNA repair protein expression profiles. We have studied through Western blot analysis. Coming to the results, so first we have developed, this is the first picture, shows the ape, shows how we developed cisplatin resistant in HND15 cells, all colorectal cancer cells, by continuous exposure, pulse exposure with cisplatin treatment. And also the figure A1 shows the IC50 determination of this. Then we have shown the figure B shows the morphological features of sensitive and resistant cells, cisplatin resistant colorectal cancer cells. We can clearly show that clearly see that the cells were morphologically different from the normal cells. And here the figure C shows the sensitive and resistant cells. The stem cell markers, expression of the stem cell markers in the sensitive, and you can see that resistant cells showed increased expression of all the stem cell markers, which confirms the resistance development. So it shows clearly that the cisplatin resistant cancer cells expressed stem cells. Hello?
Results now moving, madam. The slide earlier, same slide here, use the methodology slide.
This picture you show, because most of the whatever I was discussing were there in this picture only till now.
Okay, madam.
And stem cell markers, all the figures. That's why I was stuck with this slide for some time. Next, this slide shows the cytotoxicity assay, that is MTT assay, for IC50 determination in both sensitive as well as resistant cells with neferine and isoliensinine. We can clearly see that the neferine is showing the IC50 dose of 6.5. The IC50 dose of neferine in cisplatin sensitive cells was determined to be 6.5 minus 1.2, whereas in colorectal cancer resistant cells, it is 8.95. Actually, it is very interesting to note that in the resistant cells, with even alone, neferine alone is showing 8.9, whereas in sensitive cells, it is insulin. It is the difference is there, 5 and 12. And whereas cisplatin in resistant cells, the IC50 value was found to be 120. That means when cisplatin alone was given for cisplatin resistant cancer cells, the IC50 value is 120, whereas in sensitive cells it is only 32. That means continuous exposure, with continuous exposure, cells have become resistant to cisplatin. So the dosage required is almost 4 times. So here you can see that the isobologram, how we have shown that cisplatin concentration, IC50 value, was found in combination. And this is it shows how cisplatin with cisplatin and neferine, the dosage was, you can see here in the combinatorial regimen. Figure E shows combinatorial regimen of neferine. In combinatorial regimen, in the combinatorial regimen of neferine and cisplatin, where neferine dosage was only 4 micromolar, along with 40 micromolar of cisplatin. It induced a 50% cell death. Whereas with isoliensinine, it was 8 micromolar. 8 micromolar of isoliensinine along with cisplatin 40 micromolar induced cell death. So this is this shows the combinatorial regimen dosage also. So this figure, the cytotoxicity, this particular figure shows the IC50 determination for both sensitive as well as resistant cells, both alone as well as combinatorial regimen. Next is we have determined the ROS generation. So initially we have done the time course to find out what is the time for. So with this figure A shows the ROS generation with treatment with neferine. Here, A shows with neferine. So if at 30%, at 30 minutes, there is a peak for ROS generation. Whereas with isoliensinine, figure B shows the time the time required for ROS generation. The peak time is 3 hours. At 3 hours, the ROS generation peaked with the isoliensinine treatment. Whereas with neferine, it was only 30 minutes. So first we determined time for the ROS generation for both neferine and isoliensinine individually. Then we treated, we used NAC as inhibitor for ROS generation. With the NAC, there was no this thing, and compared to control, we always saw how much of ROS generation was there in different treatment groups. When cells were resistant cells were treated with neferine for 30 minutes, you can see that there is an increase in ROS generation, whereas with isoliensinine after 3 hours, we can see these are the resistant cells which are treated with neferine alone or treated with isoliensinine alone for different peaks. Respectively, at that for that much treatment period, we have observed the ROS generation in different these two treatment groups, and later we have tried also with NAC, that meaning N-acetylcysteine, which as an inhibitor for ROS. Again, we the same treatment groups along with neferine, we have along with NAC, we have seen that the ROS generation has significantly reduced. And here intracellular calcium levels also we have seen. Intracellular calcium levels also were significantly increased in different treatment groups, that is with the neferine treatment alone or with isoliensinine treatment alone. And here the figure B shows the combinatorial regimen. In combinatorial regimen, these two groups shows. First is control. This is neferine 4 micromolar. That means the dose which is used in combinatorial regimen alone, and this shows the dose of isoliensinine which is used in combinatorial regimen, that is 8 micromolar. And this shows the IC50 dose, that is the dose of cisplatin which is used in the combinatorial regimen. So we have shown the individual treatment groups which were used in combination, and then we can see here the combinatorial regimen. This is neferine plus 40 micromolar of cisplatin, 4 micromolar neferine and 40 micromolar cisplatin, and then this is for 8 micromolar isoliensinine, 40 micromolar of cisplatin. Because these two were the IC50 doses, no? They showed this combinatorial regimens have shown IC50 means 50% cell death. So these doses were chosen, and then this is the dose which was the IC50 dose of cisplatin alone, that is 120 micromolar. So compared to IC50 value of what do I say, the intracellular calcium level is almost in par with the IC50 dose. So the intracellular calcium levels were in the different combinatorial regimen groups were in par with the cisplatin IC50 dose. So for comparison, we have chosen IC50 dose of resistant cells also for comparison. Here you can see the mitochondrial membrane potential. So this figure A shows the neferine and isoliensinine. That means the mitochondrial membrane, the effect of this individual treatment groups, either neferine or isoliensinine alone in the resistant cells. Okay, how the chemosensitizing ability we have seen by treating with either neferine or IC50 dose of neferine or IC50 alone, isoliensinine alone, and also we compared it with CCCP. The CCCP is the positive control for mitochondrial membrane potential. So it clearly shows there is a significant decrease in the mitochondrial membrane potential. Dissipation of the membrane potential is significantly clearly observed. Compared to control, you can see that this much, so much of significant reduction in the mitopotential is observed after treatment. This is the figure B shows the combination. Initially, these two doses, first is followed by the individual dose which is used in the combinatorial regimen of isoliensinine. Then this is the dose cisplatin which is used in combinatorial regimen. So whatever doses which were used in combinatorial regimen, individual doses were also seen, but only the combination they showed the effect. You can see the significant reduction in mitochondrial membrane potential in the combinatorial regimen of neferine and cisplatin and isoliensinine and cisplatin, which are almost in par with the cisplatin IC50 dose. That is 120 micromolar. These are all cells, cisplatin. All these studies were done in cisplatin resistant colorectal cancer cells. So 120 micromolar is the IC50 dose for the cisplatin resistant cancer cells. Here, this picture shows the cell cycle analysis. Here you can see the figure A shows individual treatment groups. That means the colorectal resistant cells treated with neferine or isoliensinine alone. You can clearly see the significant subg1 peak is observed, which is indicative of the here also you can see the cell cycle analysis. Here also you can see these are all the doses which are used in combinatorial regimen. Micromolar, 8 micromolar of isoliensinine and 40 micromolar of cisplatin. These are all the doses which were used in combinatorial regimen. So individually the doses which were used in combinatorial. So, but they are not showing that much of sub G0 peak is not so much significant. Whereas here you can see in combinatorial regimen, neferine and cisplatin, there is a significant increase in the IC50. Significant increase sub-Gs. I mean, you can see the sub. So here also you can see increase in this peak in the isoliensinine and cisplatin combination, and also which is really observed in the IC50 dose of cisplatin. That is 120 micromolar. You can see that in this table he has clearly shown the sub G1 in control only 11.0. Neferine alone is only 10. You can see percentage here. You can almost 50% is given for neferine and cisplatin 33, isoliensinine and cisplatin 41, and IC50 of cisplatin is 48. Significant increase in the apoptosis in that combinatorial regimen, which is almost par with the IC50 dose of cisplatin. Here, this is the picture which shows the apoptotic features through analysis, scanning microscope. 6.5. This is IC50 dose of neferine alone. This is the IC50 dose of isoliensinine with resistant cells. You can clearly see the membrane blebbing and cell shrinkage. And here, the figure B shows the combinatorial regimen treatments. The neferine is this is only control with any treatment. This is the dosage of neferine. This is isoliensinine plus cisplatin. That means 4 micromolar. This is the neferine alone. The picture is isoliensinine alone, and this is CDDP alone. So these are all the doses which were used in combinatorial regimen. Here, this picture shows the membrane blebbing and all that. This is neferine plus CDDP. This is neferine plus isoliensinine plus CDDP, that is cisplatin. This is IC50 dose of cisplatin. So they clearly show the apoptotic features, membrane blebbing. And this figure shows the early apoptosis induction through annexin FITC staining. So here figure A shows this is the control, and here it is IC50 of neferine with resistant cells. And this is the isoliensinine. Here you can see the increased, increase in 50% almost in the lower quarter. No? This is it shows the apoptotic cells. And here also you can see isoliensinine treatment also increased. And this is okay. This is 4 micromolar dose, which is used in combination. This is 8 micromolar, which is so much significant here. Whereas in here in the combinatorial regimen, you can see the significant increase here, 30.36 apoptosis. Here also 47. Here it is again 46. This is IC50 dose of cisplatin. These two are a combination, neferine and isoliensinine in combination with 40 micromolar of cisplatin. So it clearly shows that there is a significant increase in the apoptosis in combinatorial regimens. In figure B shows this is early apoptotic population. This is here with neferine. It is 40%. Here it is 45%. So now here we have seen in the expression profile of all the apoptotic proteins, which are cytochrome C, BCL2 BAX, space 3, part 3. AKT, M2 and PI3K, these are the proteins which are involved in cell survival. So here we can see that this is pro-apoptotic protein, cytochrome C. A significant increase is observed in the combinatorial regimen. These are all the expression profiles in combinatorial regimens. You can see here, no, not combinatorial regimen. This is individual treatment. This is the sensitive cell. Then this is the resistant cell. Next, second one is the resistant cells combination with neferine and isoliensinine. The resistant cells treatment with neferine. This resistant cells treatment with isoliensinine. Then it is this is the resistant cells treatment with isoliensinine. This is resistant with neferine. Okay, this is so here you can see that there is a increase in the apoptosis. So these are the resistant cells. Okay, now you can see the ARCC1 is treatment either neferine or isoliensinine. There is a significant decrease in the ERCC1, and also BCL2. There is a this is an anti-apoptotic protein, so it's significantly increased here. Is significantly decreased in this after treatment with neferine or isoliensinine. BAX is again pro-apoptotic, so its level is significantly increased in this treatment groups compared to controls. And caspase 3 also the level is increased. And also PARP cleavage is, the cleavage is the lower level. You can see that it is increased here. That is, the cleavage is clearly observed with treatment groups. And these are cell survival proteins, PI3K, AKT, and mTOR. So they are significantly decreased. Phospho-AKT is significantly increased here. So all these things are indicative that cell proliferation is decreased. There is a DNA slightly inhibited, and apoptosis is induced. And here again, these are all the things studies with resistant cells with combinatorial regimen. Resistant cells treated with combinatorial regimen, and here you can see the CL2 is not so much affected, but in combinatorial regimen and IC50 dose of cisplatin, it is significantly decreased, and cytochrome C also the same. Caspase 3 it's significantly increased, and also cytochrome C it is increased. BCL2 is anti-apoptotic, so it is decreased. With other things, it is increased. And coming to PI3K AKT, phospho-AKT, there is a decrease. PI3K is also decreased, and mTOR is also decreased. So these are cell survival protein levels are decreased. PARP is it's not clear. PARP is also slightly increased. Here, this shows the picture of women. These are all the RNA, mRNA levels observed of all the apoptotic proteins and cell survival proteins, which were the cell survival proteins were significantly decreased, whereas pro-apoptotic proteins were significantly increased. This is cytochrome C, which I mean cytochrome C. Then this is PARP. PARP also increased in combinatorial regimen, and here this is cytochrome C, which is also a pro-apoptotic. This is also caspary, so this is also pro-apoptotic. They're all significant. mRNA levels were increased in the treatment combinatorial regimen, whereas BCL2 is anti-apoptotic protein whose levels are decreased significantly. Our mRNA levels were significantly decreased. And coming to the cell survival proteins, their level was significantly decreased. mTOR, PI3K, AKT, their levels were significantly decreased in the combinatorial regimen compared to individual treatment groups. Coming to the summary, so the cancer stem cells were developed using the pulse exposure of cisplatin to parental cells, HCT15 cells, which were acquired from the NCCS Pune. And then we have tried neferine or isoliensinine treatment alone. The IC50 dose was determined, and then we have tried the cytotoxicity. The cytotoxicity using the MTTSA, we have determined the IC50 doses. For the individually, the treatment with the neferine, 6.5 micromolar treatment of neferine was found to be the IC50 dose for chemosensitizing ability, which shows the chemosensitizing ability, and 12.5 micromolar is the IC50 dose for isoliensinine against the resistant cells, and IC50 dose of cisplatin in cisplatin resistant cells was found to be in 120 micromolar concentration in combinatorial, whereas in combinatorial regimen 40 micromolar of cisplatin was used in combination with either 4 micromolar of neferine or 8 micromolar of isoliensinine for all the combinatorial studies, and they showed the synergistic effect from the isobologram. And then isoliensinine on neferine, they could confer extensive intracellular, they induced reactive oxygen species. They induced ROS significantly in resistant cells, and also this the treatment with neferine or isoliensinine, IC50 dose of neferine or isoliensinine alone or in combination with 40 micromolar of neferine, they significantly induced decrease the mitochondrial membrane potential and increase the intracellular calcium concentration, and also they have induced apoptosis, which was confirmed from the sub-G1 peak in the flow cytometry studies, and also we could see the expression of the significant in the expression of ERCC1 in the resistant cells, and we have also seen significant decrease in the cell survival proteins and upregulation of the pro-apoptotic and downregulation of the anti-apoptotic proteins. And so this study confirms the chemosensitizing efficacy of neferine or isoliensinine, both either alone or in combination with cisplatin against cisplatin resistant cancer cells. Thank you. Thank you.
Thank you, Dr. Vijaya Padma, for the nice presentation that you gave. If anyone have any questions or doubts regarding her presentation, please ask.